super excited to be here talking to you today. But first, I'd like to acknowledge the land that we're on. So I'd just like to acknowledge the Bedegal people, who are the traditional custodians of this land on which we're meeting. I would also like to pay my respects to elders past and present, and extend that respect to any Aboriginal or Torres Strait Islander peoples here today. So now, before we get started, I'm just going to explain a little bit of the rundown for tonight. We're going to start with some presentations from the iGEM teams and the BIOMOD team that we have here before we go into a panel. There'll be an opportunity for Q&A, and where you can send your questions to will appear on the slides. And then we're going to wrap it up with, there'll be refreshments, so there's drinks and food outside afterwards as well. So, but first things first, we're actually going to begin with a short 10-question survey. So if you could pop that link into your phone or your browser, we'd love if you could complete this survey for us. It's completely anonymous, but we just want to hear your thoughts. Thanks, guys. So thanks, guys. Hopefully you had some interesting answers to those questions. But just in case you weren't sure what synthetic biology actually is, I thought I'd start by prefacing today with one of my favourite definitions of what synthetic biology is, which I did pull from Nature. So Nature defines synthetic biology as the design and construction of new biological parts, devices and systems, and the redesign of existing natural biological systems for useful purposes. And I love how this is so broad, and we're going to see that a little bit tonight when we've got 3 teams doing some stuff across a whole broad range of synthetic biology. But first we're going to start with one of the iGEM teams. We're going to start with the UNSW iGEM team, Assemblase, and they are doing a competition that's going to send them to Boston at the end of the year to present their research. But today you're just going to get a little sneak peek of some of the stuff that they've been doing over the past couple of months. So over to Beck and Toby. Yeah. Hey guys. So you're going to click the first one. As Megan mentioned,
I'm Beck, and this is Toby, and we're representing the Assemblase team this year. And I think the last time that I was speaking to an audience this size would have been back in high school English, perhaps, and it was probably about a topic that was highly insignificant. So I'm really happy today to be here talking about something that I'm actually passionate about and that all our team has worked really hard for. Because I want to paint in your minds an idea. Ideas are the driving force behind change in our world. They shape perspectives and inspire creations. So with our mentors, supervisors and collaborators, we have crafted one such idea that we think will be significant in the synthetic biology space. Our idea will act as a research tool for researchers worldwide. It will possess the potential to make prescription drugs more accessible. It will aid in the bioremediation of petrochemicals and plastics. And it will significantly reduce the cost of many industrial processes, especially in the agricultural and farming industries. But the best part about our new design is it's completely self-assembling, highly stable, and very, very adaptable. So what is this awesome new design, you may be asking yourselves? Is such a thing plausible? And if so, could these 11 weedy-looking undergraduates really have the capability to develop it? Well, it may surprise you to know that we are weedy, but we can develop it. So please join in with us as we just tell you a little bit more about our new and improved protein scaffold.
Now, when you hear the word scaffold, your mind probably jumps to some of the big construction sites over Sydney, such as James Packer's Crown Casino Hotel or the Sydney Light Rail. They're progressing every day. But I'd like to draw your mind back from that perspective on a scaffold for just a second to consider a smaller, more efficient, less light rail-esque scaffold. We're talking about a protein scaffold, capable of tethering together two proteins in close proximity to each other. Now, to explain why we need to do this and how it works, I'm first going to go over some basic science. So a chemical reaction is when a reactant forms into a product. Now, this specific process has a specific activation energy. That's an energy that the reactant has to accumulate before it's able to react forward to the product. Now, that's typically a big problem because it costs time. Now, there are enzymes which are specific proteins which decrease the activation energy, allowing the reactants to react forth into the product more readily. Now, enzymes are seen everywhere in nature. Wherever there's a reaction, you have an enzyme to complement it. Now, some examples are cell division, where there are a bunch of enzymes working together to divide the cell, digestion of food, and even antibiotic resistance. You have bacteria producing specific enzymes to digest antibiotics before they can break them down. What if we have a two-step enzymatic reaction? Then you have a reactant going through enzyme 1 to an intermediate product, then through enzyme 2 to the final product. But there are some problems with this. You see, the intermediate product can diffuse away and take a long time to come into contact with enzyme 2. It can also participate in side reactions, producing an unintended product. But we can fix this. It's been shown that if you put two enzymes in close proximity to each other, it can increase the rate of reaction by up to 77 times. Now, realistically, it's more like 2 to 5 times faster, but that is significant in the industry, and it could result in easier access to foods, to pharmaceuticals, and agricultural reagents, helping getting those products to people in need. Now, we can take this concept and try and engineer it ourselves, and that's what we've done with the protein called profoldin. Now, profoldin is produced by microbes on the floor of the Pacific Ocean near thermal vents, where water is spewed out at temperatures of up to 380 degrees Celsius. Now, the microbe that produces it is extraordinarily thermotolerant, and the protein follows suit. Now, the great thing about profoldin is that its alpha and beta subunits, when in solution, form into this 6-membered hexamer with legs extending every direction outwards. Now, we've taken this hexamer and we've engineered it so that we can hold enzymes on each of the legs as such. Now, to attach the enzymes, we've used specific attachment systems called SpyTag and SpyCatcher, and SnoopTag and SnoopCatcher. Now, if we attach SpyCatcher to, say, the alpha profoldin leg, then we attach if we attach SpyCatcher to the alpha profoldin leg, and then the SpyTag to a specific enzyme, those will associate and form an isopeptide bond holding them together. Same goes with SnoopCatcher and SnoopTag to the beta profoldin leg. Now, these are orthogonal, so they're not going to cross-react with each other. So we've got our complex, but we need to prove that it works. We're doing this through an indole acetic acid production pathway, which is a plant hormone. And it's extraordinarily useful in the industry, but we're not focused on that. For now, we're using it because the enzymes are very large. And if our complex assembles fine here, then we know it will work well in other instances. We're going to talk about some of those other instances.
So yeah, as I hope you're now starting to see, it's really evident that our scaffold is very modular and very adaptable, as Toby has hopefully emphasised. And as Toby has also mentioned, our scaffold will work with IAA production using large enzymes, and if we can show that, we can then move on to enzymes and pathways that are maybe more relevant to issues in our world. For example, the production of drug synthesis, and in specific, the production of Taxol. Taxol is an anti-cancer drug that is being used widely in industry, but there's a big problem with it. Producing its side chain is somewhat hard because it forms in two different conformations. Namely, one of these conformations is very good at being an anti-cancer drug, but the other one can have potential harms. So in industry, there's a lot of waste as they try and get rid of 50% almost of the solutions they produce. So this is where we can come along with our scaffold and specific biocatalytic enzymes to produce Taxol and its side chain, but only the structure that we want. This can be extrapolated to many other drug synthesis pathways, because by reducing wastage such as this, we can reduce price for pharmaceutical companies, which travels down to reducing costs for consumers like us. So it's evident that drug synthesis is a big way in which our scaffold could have implications in the future. But our excitement about this modular tool has extended us to look in other applications and other fields in which our scaffold could have an effect. For example, the bioremediation area, and in particular, the degradation of plastics. We are all aware of the negative effects of the inadequate plastic disposal on our health and our environment. As a plastic-free society is far from being a reality, and plastic waste builds up in our landfills and our seas, there is increasing need to find ways to combat this pollution. That's where our scaffold can step in again. With enzymes that have been proven to break down plastic, namely PETase and METase. These enzymes are very suited to our scaffold, as they are involved in a two-step hydrolysis process. By tethering them to our scaffold, we could effectively break down currently non-biodegradable plastics. This could have large benefits on our environment as we help degrade plastics. So if plastic degradation wasn't enough, we're going further. We're also looking at enzymes that could be involved in the bioremediation of hydrocarbons, and we're also then looking into industry and seeing how we could help synthesize the currently expensive pink pigment called astaxanthin. If we can synthesize it more efficiently for a reduced cost, you could be having cheap lobster tonight for dinner. So you can obviously see that there are many applications that we are exploring with our scaffold, but we are constantly finding more by talking to audiences like you. So please, after this discussion, feel free to come up and discuss these applications more. We'd probably be more comfortable than standing up here talking about them, and give us any suggestions that you have. Because it's by sharing ideas like ours with audiences like you that we can work to finding the puzzle pieces that act as innovative solutions to help the wider issues in our world. So thank you for supporting us on this journey, and we hope to talk more in the future. Thanks.
We'll now be hearing from the BIOMOD team. So they are also from another competition. BIOMOD's a little bit different, it's a little bit more focused on DNA as opposed to protein, but we're really looking forward to hearing from them.
Hey everyone, my name's Claudia. I'm from the University of Sydney, and
I'm Joel from the University of New South Wales.
And together with 6 other undergraduate students, we make up the University of Sydney BIOMOD team for 2018. And so we've named ourselves the DNAliens. So yeah, as the introduction mentioned, BIOMOD is a little bit different to iGEM. What BIOMOD focuses on is actually repurposing biomolecules like DNA, RNA, proteins, lipids and carbohydrates into more useful things. So we're less about cell systems and redesigning them and more about taking things from biology and using them in cool and different ways. Yeah. And so as part of this really interdisciplinary project, there's a lot of scope as to where we could take this project, with the only requirement being that we use biomolecules to design our devices. So what we tried to do with our project was tackle a really big issue that's ubiquitous in society, and that is antimicrobial resistance. So you may have heard of this. It's been featured very widely in newspapers. And the reason for this is that there are some really scary statistics out there about antimicrobial resistance. In fact, in 2016, the World Health Organization reported that there were nearly 500,000 cases of multi-drug resistant tuberculosis. And that's just tuberculosis alone. So think about all the other infectious diseases that are out there that our current treatments are no longer effective for. However, there is some good news. With the World Health Organization and UN taking antimicrobial resistance as a really big issue, and also other governments, a recent report from the UK government, there was a recent report from the UK government about antimicrobial resistance. And from this report, they said that if we don't change the way that we're going, by 2050, they predict that there'll be 10 million deaths per year due to antimicrobial resistance. And that, to put it into perspective, is actually more than the cancer cases out there. So it's a really big issue that we need to deal with. And as scientists, we're positioned very well to be part of this intervention. And synthetic biology can play a really big role in that. So as we mentioned earlier, this is a biomolecular design competition, and what we are using is DNA as the building blocks for our structures. So we use this using a technique called DNA origami. We take the beloved double helix that you all know from living cells, and what we do is we pull it apart. So as scientists, we don't really want to spend ages in the lab trying to design these structures. So what we do is we spend a little bit of time in the computer programming these structures to be able to self-assemble into these really cool shapes. And so what did they choose to first assemble when they built DNA origami? Smiley faces and stars, of course. So a little bit of detail as to how this actually works. What you have is a long single-stranded DNA scaffold, and you design these short staple strands which hold the structure together. And these structures actually form the DNA double helices that you normally see. And so when we go back to the other picture here, you can see that in these smiley faces and stars, it kind of has this basket weave pattern. And that is because each of those basket weaves is actually a double helix holding that structure together. So whilst these may be 2D shapes and not very applicable, we can actually use this to build 3D shapes. And that's what our project centers around, to build a nano device that can actually help combat antimicrobial resistance.
So Claudia's introduced us to this big problem and also talked about the method that we're going to utilize to attack it, which is DNA origami. The great thing about a problem such as this is we can come up with our own new creative ideas to come up with solutions that have never been done before to push the bounds of science. So the thing that we're developing is a DNA nanobot which we call the DNAlien. The DNAlien targets and destroys bacteria. We're particularly focusing on gram-negative bacteria. So the first stage of our DNAlien is to target. We want our DNAlien to attach to the surface of the bacteria. So this leads us to a major problem with antibiotics, is that they kill more than just what we're aiming to destroy. So I like to think of this as like a nuke, which destroys everything in a given radius, whereas like our approach with targeted drug delivery just tries to destroy specific parts. And the way we do this is using aptamers. Aptamers are special molecules that bind to only certain targets. And as you can see here, it's basically like a single-strand DNA that folds to form this structure, and then it is attached to specific parts. The great thing about aptamers is we can change the aptamer and we can change what we target with our structure. Once we've bound to the surface, we want our DNAlien to transform so that we can ultimately destroy the cell. So you can see here on the left, you can see here on the left an example of something that we're basing our design around that goes from being attached to the surface to embedding and creating a nanopore in the cell. So we've been brainstorming and coming up with different ideas about how we might destroy the cell. And one such idea that we've come up with, we like to call the Hoberman circle. So once our DNAlien is embedded in the cell, this structure, which is attached to our DNAlien, will expand and become larger and larger to create a big hole in the cell. And eventually, this will make the hole larger and larger and larger until this will lead to destruction of the cell. And we hope with the collective efforts of all our DNAliens that we should be able to destroy the cell. So we're really excited. We're working very hard over the next few months to continue the design and in the lab also, and we get to present our work in 2 months' time at a big jamboree in San Francisco. So if you're interested in finding more about our progress, you can follow us on Facebook, the University of Sydney BIOMOD team, or on Twitter at USyd BIOMOD. And before we finish, I'd like to thank our sponsors who've provided us with the funding that lets us do this science and participate in this cool competition. Thanks all for listening, and I hope that we've helped you show another thing that we can do with synthetic biology.
Thanks guys, that was really interesting. And now we're going to hear from another iGEM team. We're going to hear from the Macquarie iGEM team, and they're doing something similar to the UNSW iGEM team in that they're in the same competition, but they're going to focus on chlorophyll. So.
Good evening, everyone. I'd like to start off by thanking the UNSW iGEM team for all of the effort that they've put into making tonight possible. My name is Renee, and this is Angus, Carl, and Irene, and we're representing the Macquarie University's 2018 iGEM team. Our project is called ChIVes, but it stands for Chlorophyll-Induced Vesicles. As part of this project, we aim to implement and manipulate the chlorophyll biosynthesis pathway in E. coli to create vesicles. This opens up a whole new avenue for the medical field and for food industries, as these vesicles provide a space to perform otherwise cytotoxic reactions in high efficiency within the cell. This novel system of artificial compartmentalization is certain to have a profound impact on industry. As such, we will first incorporate chlorophyll A into E. coli and aim to produce these molecular compartments that we dub ChIVes, the first of its time. The Macquarie University iGEM team has done very well in the past 9 years. This year, we have had a team of 13 high-achieving science students from a variety of studies. We've got people who are majoring in medical science, commerce, chemistry, biomolecular, education, and law. We also have students who already have a background in graphic design and coding.
So, what exactly is it that we're doing? Well, we are trying to create an innovative tool which will benefit many scientists using synthetic biology. In science, microorganisms are used as hosts to produce compounds such as pharmaceuticals, which are then purified. The issue with current methods is that the products can be toxic to the microorganism as they are not normally expressed within the cell. As you can imagine, it is not possible for a cell to survive if it generates something that's toxic to it. So, as demonstrated in the first point there, this means that the cells that you would be obtaining your product from will die during this process, dramatically lowering the yields from whatever process you're trying to undergo. Additionally, more complex biological pathways typically have very low efficiencies within a cell because of a large distance between different components of the pathway, so between enzyme and substrate. And finally, the protein purification, so the last step there, is currently the most expensive step in recombinantly producing proteins, especially in the field of therapeutics, since such a high level of quality is required to pass quality controls. So, our team has come up with an innovative approach, so this bit here, to create cellular compartments in E. coli. These nanocompartments can be useful in numerous ways, such as purification and production of drug molecules, and increasing enzyme efficiency by decreasing the distance between the different components. Even if the products we create are toxic to E. coli, by creating them inside these vesicles, we prevent destruction of the cell through the process of trying to make these compounds. Alright, so how exactly are we doing this? So, we basically hijack the chlorophyll biosynthesis pathway from an algae, and we express it within E. coli. So the protoporphyrin 9, so the furthest molecule there, is already produced by E. coli, and we use it as a precursor for this whole pathway. A key step in this synthesis pathway involves the light-catalyzed reaction using protochlorophyllide oxide reductase, also known as POR, which is an enzyme, to convert the protochlorophylide into chlorophyllide. It has been shown that when cells are grown up in the dark, expressing this pathway, in the absence of light, protochlorophylide accumulates and forms semi-crystalline structures. So these structures are known as prolamellar bodies and are mainly recruited by a complex of protochlorophyllide, POR, so the oxide reductase, and NADPH. And upon exposure to light, these prolamellar bodies actually disperse and produce vesicles within the cell. And that's basically where we put our drugs and all the production and stuff. Alright, so.
Cool. So the next part of this presentation isn't quite as exciting as the stuff we've been talking about vesicles and proteins. It's about the requirements of the iGEM competition, but they are actually really cool when you get into them. So the first one we're going to talk about is human practices. And basically, human practices is a key component of the iGEM competition where teams consider how the world will impact their project and how their project makes a mark on the world. This component is all about engaging with communities, opening dialogues that assess the needs of the world, and in turn gathering ideas and feedback that shape the project. So, many teams will engage in discussions of ethics, or seek to educate the public, or propose policy that advances scientific fields. But this year, our team is kind of taking a new approach on human practices by formulating a framework for customer discovery that can be used by all research teams around the world in years to come. So, customer discovery is more of a method than anything else. It's about finding a product's potential market and illuminating current issues. For example, by interviewing professionals either in academics or industry, and finding out what problems they face and trying to suit your product for them, so you have a customer base. The material that we're going to be developing, and we are developing, is adapted from CSIRO's On Prime program. And it's going to be informed by our own theoretical development, but also our own actual customer discovery and tests and investigations. This framework will enable research groups around the globe to discover their market, which enables them to refine ideas, shape their projects to real needs, and find communities that benefit from all their endeavors, which is something that we, as part of iGEM, are trying to do with our own project. Then we go on to collaboration. Collaboration is really a big part of synthetic biology, which is one of the most open fields in science, that we're encouraged to talk with other teams, build upon our own resources. As our professors always remind us that we are standing on the shoulders of giants, and that we should always be helping other people out to grow the field as a whole. So collaboration encourages communication and cooperation worldwide between different teams. It seeks to help us aid and contribute to other teams, and also makes us be helped by them. It's a two-way process. So we are currently working with Cardiff University and the Hebrew University in Jerusalem, and we're already in contact with other teams, such as Monterrey University in Mexico, or the Hong Kong University. And so we're looking at expanding our own horizons and building good relations within the scientific community, specifically with synthetic biology. But it's not only that field that we're looking to build good relations with.
Like every synthetic biology project, public engagement and awareness is essential to both educate and find potential community-based problems in relation to our project. Last Saturday, we showcased our iGEM project in the world of synthetic biology at the Macquarie University Open Day to future students and families. This, combined with our science, safety, poetry, will not only educate the community on synthetic biology, iGEM and lab safety, it will engage the community to be more involved in synthetic biology projects we see and develop today. We aim for our vesicles to open up whole new avenues for the medical field and other industries. This will be continued through our research with the addition of vital components of vesicle formation, such as lutein biosynthesis, to be added into our plasmid. This success will hopefully lead to the ability to assemble the entire chlorophyll pathway into a single plasmid. Finally, this will lead to our ultimate goal to our revolutionized biomolecular compartments to be helping the ease of protein purification and the boosted production of medicine in the pharmaceutical industry. On behalf of the Macquarie iGEM team, I would like to thank UNSW for this opportunity to talk about our project. If anyone has any questions about our project, feel free to talk to us, the ChIVes, as we like to call each other, and we'll be hanging around afterwards to answer them. And if you want to stay connected with our journey with iGEM and our road to the Boston Jamboree later this year, please follow us on our social media. We have Twitter, Instagram, and Facebook. Thanks.
Thanks, guys. So that brings us to the end of our presentations section for today. But what we are going to do now is we're going to move on to our panel session. So I'm going to introduce our panellists, but first let's welcome them, and they're going to come and take a seat here at the front. I'm just going to explain to you how this is going to work. So the first thing we're going to start off with is I'm going to introduce all of our panellists, and we've got a few questions to kind of get the discussion going, but then we want to hear from you guys. So basically, you can send questions. There'll be there's our Instagram and our Facebook.
Is there a marker right on the board?
Yeah, there's a marker. So maybe send them to the link that will be written up on the board in a second, and we'll make sure that we get to them. But you're also very welcome to check your hand up, ask a question. Really super informal in terms of we want to hear all of your opinions, and we're really excited to hear from our four quite diverse panelists in terms of experience. We've got some industry, some academia, so let me start by introducing them. So we might start at the furthest end of the table. So this is Dr. Dominic Glover. He's actually one of UNSW iGEM's supervisors for this year. And so Dominic is a protein engineer and synthetic biologist. He graduated with a PhD in biochem from Monash, and subsequently conducted postdoc research at UC Berkeley for the development of self-assembling protein templates. In 2017, he joined the School of Babs here at UNSW as a senior lecturer in synthetic biology, and his research activities seek to understand and exploit the remarkable fidelity and precision of protein self-assembly for biotechnological applications. So we might move down, and so we have Hugh, So Hugh works for the New South Wales Department of Primary Industries, and his role is in two parts. He is at Macquarie's Synthetic Biology Research. He's in Macquarie University Synthetic Biology Research Group, and he's tasked with building chromosome 16 of the synthetic yeast genome as part of the Yeast 2.0 project. Hugh's also tasked by DPI with identifying research projects with applications to primary industries, such as forestry, agriculture, and fisheries. So Hugh completed his PhD in France at the Université Marseille in France and the University of Sydney in Australia in 2015. So we move down the table and now we have Mathew. So Mathew is an Australian Research Council Future Fellow, a CI with the ARC Centre of Excellence in Convergent Bio-Nanoscience and Technology, and a member of the UNSW Environmental Humanities Group. And he's previously been part of the Centre for the Study of Environmental Change at Lancaster University. Most recently, he held a Research Council's UK Fellowship at the Institute of Hazard, Risk and Resilience at Durham University. But Mathew's current work is focused on the social and political dimensions of technological and environmental change, on the development of negative emission strategies to anthropogenic climatic change. He's published widely on the ways in which the development of novel and emerging technologies is entangled with the profound social, ethical, and normative questions. And our final panellist is Carl. So Carl's speciality is in diagnostic products, both locally and globally. He's worked in senior roles both here and in the US, part of which included obtaining FDA clearance and commercially releasing the first diagnostic for West Nile virus. He's been the VP of Sales and Marketing for Focus Diagnostics, which included Carl leading the commercialization of the first molecular H1N1 test. And since 2012, Carl's been back here in Australia, working as the Chief Business Officer at Benetech Biopharma, where he was responsible for the commercialization and business development of their gene silencing therapeutic platform. And he's now working with several Aussie biotechs, companies on their commercialization strategies, particularly in the diagnostic space. Now, enough from me, over to our panellists. I'll pass this mic down to you, but I think we're probably small enough in here that you could just probably talk. You may not need the microphone. And we're just going to start with one of the questions that was actually on the survey, which was, in one sentence, how would you define synthetic biology? So I might just pass that down to you guys, and then...
It'd be really great if you started with someone else. Can you hear us okay?
Yeah.
Okay. I might just bring that.
It's like a snake.
Okay,
Well, there are two PhD scientists sitting to my right. I'm hoping one of you can answer that question.
So I guess what is SynBio really depends on who you ask. So there is a classic definition of building with some sort of biological molecule, whether that's DNA or protein or other materials. But that's a very broad definition, and humans have been building with biological molecules for really the past 100 years. And when you consider the fact we've been engineering organisms to ferment alcohol for thousands of years, I guess we've been doing SynBio for really a very long time. But I guess SynBio, to me anyway, is not simply building with biology, but it's how we do it. It's the process itself. So that could involve, for example, this design-build paradigm that's often described in SynBio, where you make something, test it, learn from that, and repeat the process. Sort of how you might prototype something in your tool shed, for example. And the other idea is SynBio is being able to make things quicker and easier. So developing methods that enable us to very rapidly build using biology in very reliable ways. So we have new tools and methods that enable us to very quickly sequence DNA and build new things from that DNA, for example. And so it's really those three aspects that, to me, define SynBio. But I'm sure there are other definitions at this table, and I'd be happy to hear some of those.
Yeah, sure, okay, I'll say something. So, look, if you look back in about 50 or 60 years ago, you know, there was biology and chemistry that were pretty well-defined fields. And as we sort of got a lot more understanding about the nature of DNA and move forwards, you know, you get this kind of progression of chemistry, and you get the growth of biochemistry into quite a big thing, and then you get the development of molecular biology, and these things become more mainstream. And I kind of see synthetic biology essentially as being an evolution of the field of molecular biology moving forwards, whilst at the same time being distinct in some vaguely difficult to define way. But the way sometimes I describe it to, for instance, my parents, is we had electronics, and somehow we had a transition where electronics became super cheap, and we became really, really good with them, and then it became computing. Now DNA is super cheap, sequencing is incredibly cheap, and we are now able to approach DNA science in a way that was not necessarily available even 10 or 15 years ago. That means synthetic biology is now mainstream science, and it's becoming a thing.
I can jump in this as well. I guess I cover this from the perspective of the social sciences. I'm a social scientist. And you might have heard, just in my bio, I'm associated with the ARC Centre in bio-nanoscience. So one of the questions I'm always asking is, what's the difference between bio-nanoscience and synthetic biology? And I think it depends if you start with the bio, if you start with the nano. So let's do a quick survey. Who in this room would see themselves as a chemist, by and large? Okay, a few chemists. And what about bio people? Okay, and who else have we got in the room? We got anybody else? Engineers? A few engineers. What about computer tech people? Okay. Any social scientists? No, one. Great. Okay, cool. So you know what that tells you is synthetic biology is to some degree a melange of those disciplines. And it's, in a sense, quite an old idea. It comes out of synthetic chemistry. It was developed in the early 1900s in the field of synthetic chemistry. So the terminology of synthetic biology sort of somewhat evokes that history. But if you think about the history then of molecular biology or molecular science, we're living in the shadow of the DNA revolution that didn't happen. In a sense that we thought that the discovery of DNA would herald this new green revolution, and of course that hasn't really happened in a tangible way. So we're thinking about different kind of approaches to that. So my sense is, coming at this from a social perspective, that the kind of the holy grail of where we are in bio-nano and synthetic biology is the interface between the organic world and the inorganic world, which is basically between chemistry and biology. So we heard a little bit about pharmaceutical development. So that's really about thinking about the relationship between inorganic materials and their biological reception, which is at the heart of the idea of targeting. So if we can solve that problem, we can solve the problem of targeting. It's obviously very challenging. So for me, synthetic biology is trying to get at that broad challenge. That's interesting. I'm going to come at it from a different angle. I guess my principal concern is, I've heard some elegant work here this afternoon from all three speakers, all three groups. At the end of the day, you have to be able to use it for something. What's the application? In fact, if I could be crude enough, how are you going to sell it and make money out of it? How will it benefit mankind? I mean, I think the first panellist mentioned that we've actually been doing synthetic bio for a long time. In fact, who knows what the first alcoholic beverage ever was fermented? Does anyone know? Me, correct. Approximately when?
Yeah, 18,000 years ago.
Correct, in the time of Beowulf in Scandinavia. My point is, though it had an application, it had a need, it had a demand, it had a market. So in many respects, that synthetic step that caused the creation of it fulfilled a need that was defined and identified. So that would actually be my thing to it, which is great when you get to speak fourth after everyone else.
So we might take one of the questions that kind of come through. So what are your thoughts of on the importance of educating the public as SynBio grows as a field. And I guess we can kind of expand that to be a bit more broad about the importance of educating the public on what science, I guess, how it benefits them as well. So I might throw that back over to you guys if anyone has any preliminary thoughts, and then we'll kind of move through.
It's almost impossible today. I mean, how do you not inform the public? Our access to information today is greater than it has ever been in mankind's history. So pretending that we can't provide that access to the public is actually would be ludicrous. But I think it's important that we understand the messaging. What is it we're trying to say about what it is we're trying to do? There is natural scepticism I guess, in the general population about anything that's related to genetic modification. Unfortunately, that's largely unfounded, but nevertheless we have to deal with it. So science education will overcome that. People like you moving into the community, talking about what it is you're doing, you can help sell that message and understand.
I think that there were lessons learned in the 90s with companies like Monsanto that were generally, I guess, well-intentioned in trying to be early adopters of new technology, particularly gene technology, but weren't necessarily, doing their due diligence in terms of community consultation and things like that. And looking back, no one wants to get the bad PR that these guys have generated for themselves. So certainly, Yeast 2.0, which is the project I'm working on, as an international consortium, we have on our panels and our big executive team kind of thing, bioethicists such as Deborah Matthews at the University of Washington and Jane Calvert at the University of Edinburgh. And I know, especially in the United Kingdom, a lot of grants just won't be granted unless you show that you're involving social scientists.
The recipient of those grants.
Yeah, fantastic. And doing a bit of, communication and just making sure that what you're doing is not completely out there and irrelevant and causing unnecessarily any potential problems.
So we've heard a little bit about the terminology of human practices. So I think it's worth, for those of you for whom that terminology is a bit unfamiliar, rehearsing some of the thinking behind where that terminology has come from. So we used to think about the relationship between science and society in a relatively linear sense. We call that ethical consequentialism, if you're taking notes. So that's the idea that science does things over here and it has effects over here, and then we do kind of ethical, social, regulatory analysis on the impacts of science on society, on the consequences of science. And over the last 25 years, we've realised that paradigm is quite deficient in a number of areas, and so we've tried to think about a more integrated approach. So some people talk about this as moving the side of sociological analysis upstream, so into the development process itself, rather than waiting until products have developed and then doing your bioethics. So in lots of ways, what we're engaged in the terminology of human practices is trying to get at how is it possible to have conversations about the societal dimensions of technology at an earlier stage. Well, what that means is we need to think about notions of education quite differently, because we used to think about kind of public kind of engagement or public communications through what's been described as a deficit model. The idea that publics are broadly ignorant of science, and so we need to kind of address that ignorance and thereby improve their understanding and improve their appreciation of science. The sociological literature is actually really mixed, actually. The more we tell people about science, sometimes they get more concerned rather than more comforted. So actually, there's actually a really complex sociological problem going on there. And actually, I think the terminology of human practices is trying to get us to engage in a more nuanced and a more multi-directional dialogue, rather than just telling publics about science. We actually need to engage with publics and bring them into the process of research in a broad sense. And that's what human practices, I think, is getting us to think about.
So, sorry, just freezing the screen. So I guess we're hearing about the education and kind of how that impacts on the public. Why don't we take one of the questions from up here about commercialising biotech? How does the commercialisation tie into the education and the perception that the public has of synthetic biology, for example?
So, I mean, I'm happy to make a start on that. I think what I'd like to position this as, not so much what are the barriers, but what are the 4 sort of key things, at least in my view, that you need. And I touched on one of them when we talked about alcohol and need. There's not a focus here. And that is understanding what the market need is. And within that generic title is a whole bunch of other things. Can the community afford it? Should it afford it? And a whole series of questions about that. And in fact, at a business level, you need to exquisitely understand what the value of that is, because someone has to fund the thing at the end of the day, whether it's a private company, or whether it's a government, or whether it's an insurer. Someone's going to have to pay. So understanding what the market opportunity is and being able to define it is fundamentally important. The second thing is, again, around value, is intellectual property. At the end of the day, the thing that you invent has novelty. If it's not able to be effectively patented, then you have to rely on proprietary knowledge. And although there have been some successes there with Coca-Cola, for example, fundamentally, if you've never developed something with novelty, you should be able to file patents about it. And that's the second thing. The third thing is understand what the competitive landscape is. And that's important from the point of view of where you will ultimately position whatever this particular thing is. Who else is working on the area? The lead time, particularly in the therapeutic biotechnology space, is vast. It takes years and years and many, many, many millions of dollars to bring an effective therapeutic into the market. So you better understand what the competitive landscape looks like. And an important part of that is ultimately biotech startups, like the sorts of things we're talking about, will ultimately probably not take that product into the market themselves. They will probably, what we call, create a liquidity event, do an IPO, be acquired, license it or something like that. But it's fundamentally important that that acquirer, licensee, or whoever that is, knows that you understood the access to the market. How would you get it there? What is it going to look like? What will that cost? If you haven't thought that through, they're not going to think it through for you. And I guess the fourth most important thing is make sure you've got the right people in the organisation to take the thing on.
I'll have a go at that as well. I mean, my little gag is my best liquidity moments happen in the pub.
Mine too. Yeah. And here we have this alcohol theme again. Some other fermented liquor.
Look, I'm not sure whether we still have a government today, but for those of you who are watching some of the kind of broader public conversations about this, it's been striking the degree to which the innovation language that has certainly been kind of part of our political discourse actually hasn't really generated traction for a broader audience. And I actually think that's a real problem for how we think about the kind of processes you've just been talking about. So one of the challenges I think we've got before us is how do we craft a narrative that talks about what innovation means, more than just smartphones in Surrey Hills, but that something a little bit more substantial and a little bit more systematic. Take for example the debate over precision medicine or targeted therapies that's going on at the moment. That to me, and I'm interested in your thoughts, but that to me presents some really unique challenges for how you develop a marketable proposition for actually what is quite a small disease cohort.
Yeah.
At the same time, you've got a public debate about the My Health Record saga, where it seems that the public promise of the basic infrastructure of precision health is running up against some worries in a broader public context. So that to me says we've got some really significant challenges here.
So just a moment on that, on the precision medicine thing, I think that's a really good point. I recently attended a prostate cancer precision medicine meeting in Boston, and the takeaways from that are one of the companies I'm involved with has a biomarker test for prostate cancer that is a reflex from when you're at PSA. So PSA, as you may know, for men, prostate cancer is one of the biggest cancers that kills men. And PSA is a well-ingrained pathology test for prostate cancer. It's very sensitive. It doesn't get very many wrong in terms of cancer. It's unfortunately not very specific. Approximately 50% of men who present with an elevated PSA and go on for a biopsy, which is typically a transrectal biopsy, which is a fairly invasive procedure, half of them will not have cancer. And of the half that do have cancer, half of them will have an indolent or non-aggressive cancer that will not kill them. So we talk about a social dilemma here. We have a test that's now well established, and from a clinical point of view, primary care physicians know exactly what to do with the result. They send them to the urologist, and the urologist knows what to do. They do a biopsy on it. Unfortunately, the healthcare system is paying for biopsies on people, and people are suffering biopsy, for a large number of men who didn't really need one. So to your point about identifying technology, precision medicine things that can enhance and benefit what we're doing overall, that's an example of the sort of thing you can do. If you had a biomarker or an imaging assessment that enabled you to reduce the number of men going off the biopsy, then that would obviously be an advantage. Sorry, didn't mean to dominate that.
So this is I guess it's a more specific question, maybe targeted a bit towards you more, Mathew, but also free for all. What is the most important ethical issue currently facing synthetic biology that it has yet to resolve?
Just one?
Well, we could go for maybe two, but start with one.
Well, thanks very much. I guess this is a bit of Dorothy Dixer, right? Look, maybe this is a kind of career-ending kind of sort of response, but say it. I mean, my sense is that actually ethics is not quite the right terminology, and what I mean by that is all of the research that I've seen in the broader social sciences sort of tell you to some degree that the kind of public response to emerging technologies is never really quite how it's been conceptualized in ethical paradigms. So, for example, in research that I was partly involved in the UK, we studied questions around precision applications of bio-nanotechnology, in which there were fairly clear kind of ethical kind of principles around informed consent and all that sort of standard principles. Actually, the research that came out was much more about agency. It's much more about patient agency and the feeling of, do I have control over what happens in my body, or is that given over to some external agency? What was interesting about that was it wasn't predicted from a straightforward bioethical kind of paradigm. So my sense is that actually one of the challenges is actually a different kind of challenge, which is how do we develop the language of human practices in order to create a kind of platform for more interdisciplinary conversations with the public, so which is a different paradigm than saying here's a technology and here are the ethical principles and we tick those off and that's all kind of fine and everything's kind of hunky-dory. My sense is that actually the real challenge is actually that dialogue piece, which is to some degree involving a kind of set of ethical principles, but actually is a more collective problem.
So who would start that sort of conversation? I mean, who drives that?
Yeah, well, that's right. So my sense is that there are lots of different ways of doing that. But the iGEM process is one of those vehicles, right? iGEM, as a program, encourages the teams to really think carefully about human practices. This is a great initiative, and there are lots like that. And I think we should do kind of more of these sort of initiatives, right? And I think this model of conversation is something we're just beginning as a society. But I think it's healthy to not just have humanists and social scientists in their little box and biotech people in their little box and never really talking to each other, I guess is what I'd say.
I think a beginning for that, and we're just starting to see elements of it occur now, is the shared decision-making between a patient and a primary care or a specialist physician. It's still not widespread, but to some degree that does, to a very small degree, address what you're talking about, and that's a scenario where the clinician is presenting the patient with a diagnosis and some treatment options. The clinician is not necessarily going to recommend one particular course or another, so they have a dialogue about it. The clinician attempts to educate the patient about what their treatment options are, and as a consequence they make a shared decision about it. There's a lot of barriers towards being able to move forward. That one of them is bulk billing in Australia, where a clinician is barely lucky if they get 15 minutes with a patient. Putting that aside though, in more serious disease scenarios, I think it really is important that it does form part of a shared decision and it's not this oracleized clinical diagnosis of this is what you've got and this is how we're going to treat it. Here are your treatment options. Here are some of the risks about it. Here is some information for you to read. Let's work out a decision together.
It's really connected to the conversation we had before about how people are talking a lot more with ethicists and bioethics and things in big projects at the moment. We've recently published an article about the types of engineering we would need to do to generate what we call a Mars biofoundry, to engineer crops that would be able to grow on Mars and also to decontaminate the perchlorates in the Martian soil, with the idea that a lot of the technology that we would develop in that process would potentially help feed people on Earth without increasing the agricultural footprint, therefore, increasing the productivity on the arable land we're already using. So that's really cool, and we could possibly do it, I think. I think that it's a realistic goal. We could grow plants on Mars. There's been, it's been really exciting and lots of people are talking about it. The question is, do we have the ethical approval and who's got the right to give ethical approval? And, it's something that you could argue for forever and ever. Meanwhile, if we don't develop some of these technologies, it's potentially problematic when global populations are going up really high, and we've got to be careful with how we use the land that we have in terms of, we don't want to chop down the whole Amazon to grow more soybeans or, these kinds of things. So it's a really complicated thing to deal with, and I think that you're right, it's important that we sort of have these platforms where we can, work together and collaborate and understand and come to pragmatic outcomes that are not necessarily unethical.
Yeah, I mean, if you're talking about Mars and growing crops on Mars, I mean, just talk to Richard Branson, right? I mean, he's the one who gets the final say, right?
I think it's Elon Musk.
Elon Musk. I mean, my sense is that, that the agricultural context I need to think a little more carefully about, but in the medical context, you now have this interesting situation where you have, in a consulting context, a range of different bodies of expertise all happening at the same time. So you've got the medical practitioner, you might have a genomic or genetics counsellor, but you also got the patient themselves, often who are bringing lots of expertise.
And the payer.
Yeah, that's right. Yeah, absolutely. And so, that's the old model was doctor said so, and that's no longer the case. So we're just at the beginning, I think, of working that out. That we've got a really interesting set of negotiations going on in these kind of spaces about who has control over what happens to my body, or what happens to the healthcare of the nation. All these broader questions.
And can we pay for it?
And can we pay for it? That's right. Yeah. And so I don't think we've resolved those questions in any sufficient way. But I do think it's important to have these kind of discussions, especially if we're going to do that on Mars, right?
We've got one of the questions which has come through is, how do you think SynBio's open source kind of ideology is going to harmonise with some of the ideas we've been talking about commercialisation, particularly if we're talking about information and patient access and so forth? How is SynBio's open source nature, is it either going to have to change, or are we going to kind of have a new paradigm of consultation and development?
It'd be interesting to see what some of the big companies like Amyris and Ginkgo and other companies like Bayer who are already exploring the SynBio space, what they're thought about open source biology. I think that it's often touted as a tenement of synthetic biology, and I think that it's really great that iGEM is sort of a super open sort of platform and things, but the practicality of it is, in a commercial world, I don't know how real that really is.
History is not always the best way to look at what might happen in the future. But a long time ago, when I first graduated, monoclonal antibodies were going to be the silver bullet. And today, they make a major contribution. I'll tell you, that was nearly 40 years ago. So even if it starts now, and there's no reason for it to take a long time, You see all these new technologies now. People talk about CAR-T and CRISPR-Cas9 as things we were discussing before. They're going to resolve everything. I guarantee you, unless it's for an orphan indication, we will not see them in a major disease indication for probably 15 years. That's unless things change dramatically. So time ultimately is probably one of the things that I would prefer to see. Impressed when technology that clearly has a benefit, and this gets to the regulator piece, and you made the differential between regulators and ethics. I think there's a big difference. Is it ethical to give someone who's got nothing left in a cancer treatment a last gasp at it, if that's what they want to do? And I guess my answer to that is it is. But regulators, notwithstanding Australia, are a little bit different. But the United States, it's very hard to get them that drug.
I mean, it's certainly very interesting that SynBio is so open source that there are now, for example, biohacker spaces that can essentially go out and almost try to replicate generic drugs, attempting to manufacture, for example, this open insulin idea where they're effectively producing their own lab sources of insulin with the idea of ultimately getting that approved and at least used as a generic medicine. So it is interesting that people are
where the intellectual property, and that's why patent law. That's why things like having real expertise in patent filing and patent law becomes fundamentally important.
But it's also interesting that these companies like Ginkgo and that, they don't actually patent some of their ideas now because it's actually very easy to innovate around some of their modifications. So they like to keep it a bit like SpaceX does and just keep it in-house and don't actually put any of the information out there.
But I think that's because the IP space hasn't caught up enough to say, look, that's obviously a workaround.
If you change one amino acid, it's a new patent, right?
And I mean, even the US patent offices, they're not going to accept a novelty on something like that. But the problem is that there are other workarounds and they haven't caught up.
I mean, I think there are. I mean, the question that we started with was about the open source kind of ethos. I think there's real kind of analogies here with the open source software community. So all of those people now work for Microsoft, right, and Google. So presumably, all the open source bio people will end up working for major biotech, right? And that's how the industry will work. And so the distinction between closed and open is, I think, a little bit overstated. But there are some interesting kind of developments on the horizon. So look at the way universities are leading some of this discussion, and they're doing kind of cost-benefit analysis of their knowledge transfer officers and realising their knowledge transfer officers probably cost them more than it's coming back into the university via licensing. So will that mean universities adopt different patenting strategies and IP strategies? Interesting space to watch. Another interesting space to watch is the kind of economy of clinical trials. So we're kind of at the end of the blockbuster breakthrough kind of IP pathway that the pharmaceutical companies have relied upon. So what will that mean then for the future economic prospects of different kind of pathways of development in pharmaceuticals? Interesting space to watch. And what will that then do to the way IP works in this space? I think really interesting developing thing to watch.
So we might move to a question about, so what do you think individually, what do you think the greatest barrier to synthetic biology is that you think you may have a solution for? So that's a big question. So you could, why don't we start with the barriers, and then you might be able to propose solutions to each other's barriers as well. It's a loaded question.
No, it's an interesting question. It's very hypothetical, so I think it's a hypothetical answer. I mean, whatever it is, I mean, anything that's got potential novelty in the market, the single biggest barrier is adoption. Will people use it? Will they buy it? Will you change? And I have to keep speaking about clinical, because that's what my background is, but changing clinical behaviour is extraordinarily difficult. So I would say, you could have the cleverest, the best idea in the world, that actually, and turn it into a wonderful product that someone's got to buy. And that's, for me, that's one of the biggest barriers.
agriculture is certainly regarded as an early adopter of gene technology, and even there, you know, there is a lot of, I think, if it's not broke, don't fix it kind of thing. So I guess it depends on your business. Certainly, maybe not within a medical situation where it's very heavily regulated, but there's different business patterns. You know, do you become an early adopter and take that risk and reap the benefits, or do you follow the path that you're established? So how do you cope with that? I've got no idea.
Well, I was hoping no one would ask me that. Seriously.
I mean, I would approach this question like a social scientist, and I'd argue with the terms of the question, right? That's what social scientists do. Excellent. And there's two ways to think about this, right? So there are the barriers to particular outcomes of synthetic biology research. But you asked the question about synthetic biology as a field. And what we know is all fields go through a bit of a kind of hype cycle, and then they fall down the other end. And synthetic biology is kind of, I think, kind of almost touching the top of the hype cycle and is about to kind of touch over. So then how do you respond to that? I think you respond to that by being real. And I think one of the challenges we have in our societal kind of discussion of science is actually the challenge of how do we talk about not living up to the promise and actually being real and actually genuine and saying this is complex, this is difficult, we haven't really delivered what we said we were going to do, but here are the reasons why. And that's actually a really difficult conversation to have with the public, where we feel propelled to say, look, this is going to cure cancer, it's going to send people to Mars, and it's going to give us endless food to eat. That's the kind of promises attached to these fields, and we're never going to deliver on that stuff.
Well, I don't know.
Okay.
That's a lot. Okay. I mean, if you look historically over the amount of agricultural improvement that we've got through gene technology since about the 90s, I think it's within trillions of dollars, maybe, of improvement of crops and agricultural productivity. So even if the goal of getting to Mars failed and we improved photosynthesis by 2%, that would represent a major improvement in agricultural productivity. Bill Gates' foundation, I think, spent $40 million to improve, I think, a cereal crop's photosynthesis rate by 1% or something along those lines. And there's a lot of other stuff. And certainly, as we're becoming aware of how important sustainability is on a bunch of different levels, so in terms of environment, but in terms of business practices and things like that, I think that there is a lot of hope that synthetic biology will certainly deliver for that through various things like microbiological consolidated processing, etc., etc.
But the danger is that you're in this hype cycle, and so when you fall down the other end of the hype cycle, that's where the sort of public conversation starts to change, right? And so I hang out a lot in laboratories with scientists, and I do kind of what's called ethnographic research, and it's always really interesting to how different scientists talk to each other, and when they talk to the public. When they talk to the public, they feel propelled, I think, to have a much more confident stance. And actually, I think there's actually value in actually kind of being a little bit more genuine and real. I'm not saying people are lying, but they're actually sort of saying this is really complex stuff we're working on. And actually having that dialogue, I think that means you can avoid some of the inevitability of sort of the critique that, look, we're giving you all this money, and what have we got from it? I think that's what I would say.
I think the challenges to this is, I don't know what I was going to say. No, it's fine. We've got a problem connecting competencies and needs, which is something that was touched on a little bit earlier. Competencies and needs and communication. So it's sort of a three-way thing. There'll be biologists, synthetic biologists, trapped in the laboratory who've never met a farmer, never been to Wagga Wagga, and they've never been to, Griffith or any of the major New South Wales agriculture hotspots. They've never met a farmer before even. They're from, downtown Sydney. We've got a farmer who's sort of out on the farm, watched Today Tonight and learned that bacteria is terrible. And then we've got a startup company that's able to produce a new herbicide in bacteria, or wants to be able to do that. how do we connect all of that together? It's really quite a difficult scenario. So in DPI, I think I've done a lot of touring around the country to try and communicate what we're doing in DPI, in synthetic biology, also to try and connect to maybe some of the more informed sort of public about what they think about it and what they kind of need. But it's not even a one-person job sometimes. I think that it's a whole situation and it needs to be considered seriously how a synthetic biologist who's a career academic and a company that wants to produce something for a farmer who needs to communicate that to someone that can start a company, that's the barrier for me, I think.
But I think the barrier in SynBio is much lower than it would have been a few years ago in biotech, for example. If you were to start a biotech company, you probably need several $1 million, lab space, equipment. Certainly in San Francisco, there's a number of incubators that specialize in spinning out synthetic biology companies. So they give the entrepreneurs, who are often little more than undergraduates or postgrad students, some lab space, a couple $100,000, and 3 months to build a demo product.
And an option.
And an option. Yeah, exactly. They take 10%, right? But IndieBio is one of the incubators, and they've already spun out over 50 startup biotech or SynBio companies. About a third have already failed, as you would expect. But some of them have actually now raised, $10, $20 million in funds from just a very, very simple beginning. And I think that's because it's now easy to outsource a lot of this work and really build it up very quickly.
Right. Yeah. Do you want to?
Open question to the panel. Let's imagine that 4 synthetic biology startups open tomorrow. In 5 years, these 4 companies are making loads of money. What, in the opinion of the panel, would these synthetic biology companies be doing?
All 4 of them are making money?
Let's imagine all 4 of them making money. What each of you get to give a synthetic biology company, and what would you making money in 5 years?
I mean, I think the best example are these companies taking a cheap substrate like sugar and making something expensive, right? There's quite a few of these very simple startup companies that have just make a very expensive product from sugar, and they've been very successful. Ginkgo, for example, now have raised $300-400 million, and they're basically making perfumes and cosmetics from fermented microbes, basically, right?
It's hard for me to envisage, at least in the sort of medical space, of any startup making money in 4 years. Some of them I'm wondering whether they ever will. But I mean, that's a fact. That's a feature of regulatory, typically. I mean, define making money. I mean, I can think of some huge American biotech companies that still don't make a cracker in terms of income, but their market cap is $5 or $6 or $7 billion. And they have round after round of investment, and people keep investing in them, and they eventually will probably produce a product. I had a meeting in Boston recently with a company called Bluebird Bio, and they're being in the CAR T space, and I think their market cap is about $2.8 billion, and they're not making any money.
Look,
So I can't answer your question for you.
the first thing you should know is that you never take market advice from a social scientist, right? So I'm not going to answer the question because I don't know. But what I would say is we don't live in the Bay Area, right? So we don't have the same level of venture capital floating around, our environment. So the choice is: do you move to the Bay Area or do you stay in Sydney? And I'm sure that's a question that many of you are asking, right? I think while we don't have lots of things that we might want here in Sydney in terms of our innovation culture, I think Australia does have a really intriguing innovation culture that comes out of a kind of necessity. So it's interesting your linkage with the DPI, right? That's a kind of where Australia really is often quite good at kind of real world problem solving, particularly in kind of unique landscapes that characterise the Australian agricultural context or our particular kind of social context. And so we have a real history of doing that as a nation, and actually it's a really intriguing kind of innovation culture that we don't often talk about. we often talk about all the things we don't have. We don't have the startups, we don't have the venture capital, we don't have the Silicon Valley, blah, 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 blah. What we do have is a kind of practical, pragmatic innovation culture that's pretty well established now. And I think that's where I would want to position myself if I was doing this, which I'm not.
So it's what would for successful companies be? That was the question. Sorry.
Yeah. Well, what would a successful company be doing? Like, what would this synthetic biology, imaginary synthetic biology company, opens tomorrow, but in 5 years it's going to be making money? What do you imagine it does?
Well, look, I think an example of something that's really exciting that's happening in synthetic biology, sadly already started company, Eligo, based in Paris, I think headed up by maybe David Picard or someone. Microbiome engineering, it's really exciting because I know that Ginkgo and Bayer have also done microbiome engineering or are doing it to improve nitrogen fixation. They think that they can reduce nitrogen-based fertilizers by 50%, so that certainly would be a huge money spinner. So I sort of see microbiome engineering as being one hugely exciting field. I think sort of microbial cell factories, just finding something cheap, making expensive stuff out of it. That's always a pretty surefire money printer. I think being really clever and being on top of the cutting edge of technology and doing something that no one's thought of is always the way to innovate. So that's sort of me saying, I don't know. You've got to be smarter than everyone else and have that cutting edge behind you and make some magic.
I mean, I think it's all about finding a problem that you can solve, right?
Exactly. Necessity is the mother of invention.
I might take us back to something Mathew said. You said about the innovation culture in Australia. And we've had a question come through about how do we convince universities that interdisciplinary collaboration is actually really valuable? Which is an interesting question on a couple of levels. But in terms of innovation culture and interdisciplinary collaboration, why don't we start with why it's important in that conversation of trying to convince our universities to fund it.
What do you mean by interdisciplinary?
So I guess when we look at a project like, let's take iGEM or BIOMOD, we've got biology, we've got chemistry, we've got mathematics, we've got ethicists. How do we convince our universities that to invest in teams that are a little bit more well-rounded in that sense? But I guess also looking at grant applications might be.
I mean, the university seems to always have initiatives to do that. I mean, this 2025 strategy at UNSW is all about combining this, but it seems more buzzwords than actual action to me. Maybe I shouldn't say that, but that certainly comes across, and I don't see any real drive to universities to actually do that, I guess.
It does, it's that kind of vexing question of primary research, which is fundamentally important, that is not necessarily driven by an application. I know I said identify market need before you, but there needs to be some baseline foundation work that gets done, and that's the challenging part. That's probably at the very, primitive is the wrong word, at the base level. That's probably best done within a particular discipline. But then there is a grey zone as it advances a little bit where it would be appropriate to start to combine different disciplines. I mean, universities, University of New South Wales, Sydney University are huge behemoths. I mean, trying to get them to change direction must be extraordinarily challenging. But I guess they all have offices of commercialisation. I would see that as fundamentally their role.
I mean, I think it's really interesting. I mean, you mentioned some of the developments in Europe and the UK, and some of that interdisciplinary research is actually being driven by the research councils, which isn't. So when there's money attached, people will collaborate, which is interesting, right? So I think that's an interesting development. And I think we're seeing some of the signs of that in Australia, and some of the university strategies I think we all probably are aware of in our own institutions are, trying to evoke some of that ethos. I spend a lot of time with chemists and biologists, and I'm really intrigued by how disciplines that are so proximate actually really struggle to talk to each other in quite fundamental ways. Like, conceptually talk to each other. So actually there's a really deep philosophical problem that actually goes to how we educate our scientists. And so, you know, most university campuses in Australia have relatively competitive, teaching kind of setups where basically disciplines are competing for students. And the old model of a kind of liberal arts model where you do a little bit of this and a little bit of that, and you might do some humanities, and humanities people might do some science. We've sort of progressively undercut that, I think, in Australia for a long time. And actually, I think that's a huge problem. I think we need a more interdisciplinary model of education and teaching, and that could extend right into high school as well. And actually, I think some of the ways in which we kind of struggle to talk to each other can be addressed much earlier in the process, rather than waiting for the research councils or the research initiatives to be the place where that happens.
And briefly, that's actually not unique to academic institutions. That's reflected in large corporations, big pharmaceutical companies in particular, you know, where they have a drug for treating a particular disease, they have a device that could ultimately prevent the need for the use of that drug, and the two departments don't talk to each other.
I think we might take one of the questions which has come through again about, has SynBio made any major developments in relation to microbial drug resistance?
Yeah, it has.
Yes, okay, go on, next question.
There's a really awesome paper called Sequence-Specific Antimicrobials, and I think I mentioned earlier Eligo Biosciences, which is the microbiome engineering company. And this is actually one of the papers that got me interested in synthetic biology at the start, and what they did was, inside a bacteriophage, they encoded Cas9 and guide RNA for chopping an antibiotic resistance gene. And so they introduced the bacteriophage into a mouse infected with this particular multi-drug resistant bacterium, and found that the mouse still succumbed. And so then they said, well, wait, let's introduce the bacteriophage with a non-resistant bacterium as a co-infection, and found that they could clear out the resistant bacterium and replace the infection with a non-resistant bacterium, and then after the replacement of the microbial infection with a non-resistant strain, just kill it with the regular antimicrobial. And they put that out there as a novel antimicrobial, a sequence-specific one. And I thought, I've spoken to some more antimicrobial microbiologists I'm friends with, and they're like, phage therapy, not so sure. But certainly I found that that concept was a synthetic biology concept that is really exciting and promising for novel antimicrobials, and so that's why I said yes.
Might open it up to the floor if anyone wants to chuck their hand up and ask a question. Yep, Daniel?
I have the opposite question from Jan XV. He asked about what do you think a company starting tomorrow would be doing if in 5 years they make a lot of money. But what do you think is being overhyped for sure in SynBio right now? Either it's a technology or an aspect or a promise that people keep talking about, and you think that they just completely off the mark, should not be giving it any attention, any money?
Can I say something about hype?
Absolutely.
As a response to that. So I wrote an article recently, and it was about agriculture and synthetic biology. And the reason I wrote the article is because I think that about 90% of synthetic biology articles that I've read will mention agriculture as a buzzword without actually specifically mentioning any real impacts on agriculture. Are you talking about gene drives that are going to increase, make breeding happen faster? Not really. Are you talking about new antimicrobials for improved outcomes for cattle management? Or, what exact things? So I feel like kind of an answer is waffly, non-specific, using agriculture as a buzzword, synthetic biology that doesn't really do anything but says that it's doing something. Does that make sense?
It does make perfect sense.
Yeah, myself.
Yeah, I think one of the other things I see it frequently is, we have cured so many diseases in transgenic mice. It's unbelievable.
Yeah.
And, that's the media says we've got a new cure for a new cancer. And the work is preclinical work. It's fundamentally necessary. But to your point about hype is, that's the very beginning. You've demonstrated a mode of action in a transgenic mouse. It's a mouse. Okay, might have some human tissue, but still a mouse. And then there's a lot of other steps you've got to go to before you get to clinical proof of concept, which is phase 2, and about $100 million, depending on the indication, maybe more.
It wheels back straight into what you were talking about before. I mean, the more you talk something up to be, it's going to be the cure-all, the harder you're going to fall.
Yeah, absolutely. So, I mean, there's two aspects to your question. One is about hype. One is about should we give those fields more money? I'm not against giving.
And if you have any examples, that'll be great as well.
CRISPR. I think CRISPR is a perfect example of a technology that will quickly develop a more nuanced understanding of. I mean, I heard the other day someone claim that CRISPR technology will allow us to cure every disease without side effects. I mean, what an amazing...
Sorry, that was my answer.
Yeah. So if that's the kind of claim that are being used around CRISPR, that bubble's going to be popped, right?
I mean, I think the other great example, non-medical one, is so BP gave Berkeley $500 million about 5 years ago to make biofuels, right? And it was this great idea that we'll all be like, chopping trees and growing corn to make fuel to put in our cars. But of course, the oil price went down and all that research crashed. So ultimately, it all comes down to price as well. And so there are companies making synthetic meat and synthetic products, synthetic eggs and so forth. But ultimately, they have to be cheaper than, or at least equal to the current available products as well. So often it does come down to demand and price as well.
Yeah, I did my PhD on algal biodiesel.
There you go.
I've got opinions.
So we might take one or two last questions. So we'll call this one a penultimate question. If we were running this panel in 2025, so 7-ish years from now, what do you think we'd still be talking about in synthetic biology, and what do you think would be redundant? What are we moving towards? Where's the future heading for synthetic biology?
In 70, I'll be on a beach.
Look, I'm working in genome engineering, not in the CRISPR-Cas9 sort of genome engineering, but in whole genome synthesis and design. And I think JCVI 1 and 2 and these kinds of minimal genomes and whole genome design and synthesis is really exciting and fascinating. I can remember John Glass from JCVI talked at a recent conference that I was at, and he talked about how FTSZ, which is the spiral sort of proteins that sort of bud two cells into the daughter cells, that was not included in their minimal genome, and that blew my mind because, I mean, what, do the cells just kind of like magically bled apart or something? How does that even work? So, I mean, that for me is something that I don't think I'll ever stop thinking about or talking about. And hopefully yeast 2.0, which is sort of probably going to be the biggest thing that I ever get involved in my whole life, which I'm doing at the moment, hopefully that takes a similar aspect, being the eukaryotic sort of whole genome construction project, the first of its kind. So, that's what I think. And then moving from there in 2020, was it 2025?
2025, yeah.
2025, we'll be, talking about something more exciting. I know that they're talking about moving this kind of whole genome synthesis and design strategy into humans and things like that for human cell lines and cancer research and stuff. So that will certainly be something that will take a lot of time but be potentially transformative and interesting. But I think maybe something that will drop to the wayside a little bit. People claiming stuff like I overexpressed a protein in tobacco and it's the most amazing synthetic biology project. Hopefully that drops off. It's not very exciting.
Can I tell you what I hope we're talking about, rather than what I think we will be talking about? Which sort of relates to your point. I mean, I actually think one of the issues we have across these fields is this problem that people talk about as clinical translation, which is how many mice have we cured, but how many humans have we not cured? which is actually, at heart, I think about how we report results of research. So it's the kind of, I did this little thing and then I reported it, and I didn't tell you all the other parts of my experiments didn't work, and that's all kind of just hidden in the lab stuff. I think how we report what we do and how we build a culture of talking about science and talking about our research, I think needs to change, so that we actually are addressing this really big problem of clinical translation, not just as a need to just do more and more science, but it's actually to change the culture of how we talk about science. Alongside that, I think one of the areas that I'm really interested in is the gene, the organic, inorganic, and environmental discussion. So, what does it mean to think about the biological environment of precision therapies? I think that necessarily includes questions of a societal nature. So it includes questions around exposure. So what does it mean to think about our biological exposure as both a biophysical problem but also as a social problem? The fact that we live in crappy environments, basically. I think it's a good challenge.
I mean, again, I have to focus on a more specific area, and it is beginning to evolve that way. But I would like to think in another 7 or 8 years' time, there is not a single therapy that's cleared by regulators that does not have some type of companion diagnostic associated with it that gives you very, very exquisite results on it. This is one of the problems, in our clinical trial architecture now. If you get an endpoint where you had a cure rate of, 60-70%, that's probably okay, except 40% of people it didn't work on, and yet you paid for this therapy. So for me, again, this is a very narrow area. I'd love to see more targeted therapies that are applied on the basis of precision medicine that says, I tested the patient that had this disease syndrome, and the appropriate therapy for it is this, and we know it's this because that's what the test is.
Yeah, I mean, I think for me it's more really the direction the research is going in SynBio, and more about how we actually do the research itself. So there's this massive drive now, it's happening in Macquarie, of course, is introducing robotics and automation in SynBio, so we can more rapidly build very complex systems and what impact that will have. I mean, CSIRO has just announced they're also building a big robotic biofoundry now in Australia. There's now a dozen overseas, and I imagine most universities over the next few years will establish these sorts of facilities where you can very quickly design something, build it and test it, and I guess we'll be talking about how we can more rapidly build things in 7 years' time.
So are you saying that synthetic biology as a human profession is increasingly irrelevant and will all be done by robots?
So there are some of these SynBio companies where the scientists don't go into the lab anymore. They sit in their office and design everything on a computer. It goes to the robot and it all gets built and tested while they're at home sleeping.
That's not accurate, surely.
It is very accurate.
This sounds overhyped.
I know someone who doesn't go into the lab and hasn't been in for a year.
And works in Ginkgo.
Not Ginkgo, Amaris.
Okay.
So the message is, if you're sitting in the room and you're an undergraduate and you want to work in synthetic biology, get out now. Replaced by a robot. Is that right?
We're not going headed that way.
So I guess, yeah.
So, let me take a break this, because my question is very close to the question about career pathway. So I'm just starting my course at UNSW Global. I'm a new student here to UNSW. I'm going to take the foundation study first, and then I will do a bachelor degree in bioinformatics engineering. It's a dual degree combining bioinformatics engineering and biomedical engineering, and it will take me around 5 years, including the foundation. It will be 5 years. And after that, I have planned for myself a 10 or 15 career plan, and I will do another degree. So after the master, I will do a PhD in neuroinformatics and neuroengineering. Is it a good career pathway? So we are looking for about the far future, so not about 2025, or maybe for myself it could be 2030.
Well, you have to think about what you want to do with that career path. Do you want to go into research? Do you want to go into industry?
How
do you want to use that?
So I had a talk with my professor back in Vietnam, and he had also asked me about that question. Would I like to be a researcher, an entrepreneur, or just someone who invent a new technology? So I have answered that question. I would like to be an entrepreneur. So at first I will finish my degree and then I get the knowledge to build a new technology so that I will bring that technology into the industry.
So we might just save these types of questions. This is probably a really good segue point into, I'm going to ask you one more question, one word, and then, or is there a last?
Yeah, I do have a question. Do you want me to save it?
We might save those questions. We're going to keep our panellists here for a little bit. That's okay, and so you can come up, you can ask your questions, and they will be hopefully here for a little bit to answer. To answer them, we left a little bit of time in the program for you to come and ask them questions. But also there'll be refreshments outside. There's fermented beverages and foods, so please help yourself. But I guess the last question is, what is the thing you are most excited about in one or two words or a couple of words in the synthetic biology space? What's the one thing you're most excited about? Or something that you've seen that you think is really exciting. Just one thing that you think is really exciting that's happening that we might want to learn about or potentially use as a career driving point if some of us are still undergrads.
I'd like to sort of start with a generic kind of general response to that is, for me, it's sitting in a room full of people like you and seeing the three presentations that we saw this evening and the passion and the thoughtfulness that you all put into it. And quite frankly, I'm very comfortable with where the world's going, if particularly science is in your hands. So it's all up to you.
So that's concluding our presentation.